Okay, well, let's go and get rolling for today. So, several announcements. First of all, over the weekend, some of you may have seen problem set 4 scores pop up on Canvas and then maybe even disappear this morning. So we usually record those scores and then we mute the assignment. That's just, we forgot to mute the assignment. So if you didn't have one this weekend, but some of your friends did, don't worry about it. We're still grading them, okay? So all 3 TAs contribute to grading, and so one of them got done sooner than the other. The other thing about that is that there was a mistake in the key. So the score that you may have seen pop up might be different than what's going to pop up in the next couple of days, just because we caught an error in the key. So anyway. All right, so keep that in mind. Hopefully we'll have those scores out to you in the next couple of days, okay? The group work assignment, I should have, did you see group work assignment grades? Okay, good. I thought I had released those, okay? So you should see that. Problem set will come soon. Quiz 12 is due tonight, so remember to do that. We've covered definitely the material for Quiz 12, so you don't need to wait on that. Quiz 13, I would definitely wait till Friday. It's due on Friday. It's almost entirely over amino acid metabolism, which we haven't even started. We'll start today. And then problem set 5, we'll have covered the material for problem set 5 after Wednesday, okay? And that's going to be due on Friday as well, okay? And that will round out all the assignments for the semester, and then of course we have the final exam. Remember, the final exam is not going to be a cumulative exam, okay? So it will be over essentially lipid transport, lipid metabolism, and then finally amino acid metabolism, okay? I will give you the full 2 hours in the and during the final to take that. I will write it though, just like I've written the others, to get done in 50 minutes, okay? So you should have plenty of time. Won't be a real time crunch on that one. The final exam, if I remember correctly, is going to be on Tuesday at 4 o'clock. We will have it in this classroom, okay? So come here on that Tuesday of finals week, okay? All right, our riddle. So I keep stuff in. I'm usually on top. I can sit to the right, to the left, or the middle. I'm used to go pow. Can you figure me out now? I heard like cacophony. What?
Speech bubble?
No, not a speech bubble. President? No. Jack in the Box? No. There's one of them, no, 2 of them in this classroom. Huh? No? People are wearing them. No, there's 3 of them. I just saw another one. Yeah, not a hat. It's a hat, but a cap. Yeah, I keep things in. You put a cap on something, right? You can wear a cap any direction. And then also, right, how does it go pow? A cap gun. A cap goes pow. I know, some of you are shaking your head once again like, it's okay, I've only got 2 more of those. I can make it, I can do it. 2 more, okay? All right, no, that's definitely not us. We're here. Okay, so last time, this would have been Friday, we left off essentially right here on Friday. Let me go ahead and back up 1 or 2 slides here, or a couple slides, just to kind of set the stage for today. So we're talking about now the synthesis of lipids, which is actually a fairly expensive biogenesis pathway from an energy standpoint. So we mentioned that it'll take 8 acetyl-CoAs and take those and make a 16 carbon lipid. It's going to need 7 ATP, 14 NADPH to do this. That doesn't account for the ATP needed to move the acetyl-CoA out into the cytosol. So lots of energy being expended to make a 16 carbon lipid. I broke the process down into 3 steps. The first 2 we went over on Friday. So we talked about how acetyl-CoA will be transported into the cytosol as citrate. That citrate molecule will be broken down to oxoacetate and acetyl-CoA. ATP is required to do that. Okay, for every one of them, citrate lyase uses ATP to make every acetyl-CoA in the cytosol. Okay, that oxaloacetate can be converted through a couple of steps to pyruvate, and that can make some of the NADPH that's going to be needed in this process. Okay, and then we also then talked about how 7 of the acetyl-CoAs, so we go here, step 2, this step right here, 7 of those acetyl-CoAs will be converted to malonyl-CoA by the enzyme acetyl-CoA carboxylase, which is the rate-limiting step of this entire process. Okay, so what we have now sitting in the cytosol, all right, will be 7 malonyl-CoAs and 1 acetyl-CoA that can be used with NADPH to make the fatty acid, the 16-carbon fatty acid. That's what we're going to look at today, which would be step 3, catalyzed by this enzyme, fatty acid synthase. Okay, so let me go ahead and scroll to this new slide here. Okay, this is fatty acid synthase. Okay, fatty acid synthase, it also goes by the abbreviation FAS1. Okay, cytosolic enzyme. It's a big enzyme. Okay, in fact, when you look here, on the left, that would be the crystal structure of this enzyme. We're going to look at depictions more consistent with what you see on the right, which is this kind of cartoon rendering of it. There are a total of 7 domains in the enzyme. Okay, so 7 domains. All of them listed here. I do not expect you to memorize the names of these. Okay, we'll look at the function of some of these, not all of these, okay, in this process. But those are 7 domains. It is a 240 kilodalton protein, okay, 240,000 grams per mole in its molecular weight. It's big, okay, relative to other enzymes that you would find in the cell. Okay. Now, the reaction that it's going to catalyze is this. It's going to take that acetyl-CoA and the malonyl-CoA. There's 7 of those, remember. And it's going to make this 16-carbon saturated fatty acid, okay, and 7 HCO3 bicarbonates. Coenzyme A, there will be 8 total coenzyme A's that come out of this process, okay, and the process will need, as we will see, 14 NADPH. Okay, so that's the reaction that it's going to catalyze this enzyme. It's going to take. Oh yeah, question.
What is it written on the end of the thing?
You can't read that? Why not? Come on, you've been with me long enough to read that. It's HCO3 minus. And it's a bicarbonate. You could write CO2. That would be fine too. It actually comes off, I believe, as CO2. It'll be converted to HCO3, though. And there's 7 of them. Okay. So let me write this again. For those of you that are picky. There we go. Okay. There we go. Right, so that's the reaction it's going to catalyze. Now, it's going to take 7 rounds, okay, to do this. There's a lot of similarities between this and beta oxidation. Beta oxidation was this process that there's these successive rounds that it took to do that. Same thing here. It's going to take 7 rounds to make a 16-carbon molecule. Right? In those 7 rounds, there will be 6 steps each round. Okay? And we're going to look at, of course, one of the rounds, and then we'll extrapolate from there. The 6 steps to the other rounds. Okay. Now, if we come over here and we look at these 7 domains, okay, I'm going to highlight some of the ones that we're going to talk about. We're going to talk about this synthase, which catalyzes a condensation reaction. We're going to talk about this dehydratase, which catalyzes, as the name might suggest, a dehydration, not a dehydration, but it is the removal of water. I have to be careful, okay? And then these two reductases, which catalyze reduction reactions, okay? So those will be some of what we'll look at. Not awesome. That's, oh, whew. Okay. Hopefully that doesn't happen again. I better be careful. Okay. All right. So, so those are some of what we're going to look at. All right. We will also, I'm going to guess I'm highlighting pretty much everything here. We'll talk briefly about this, and we'll talk briefly about this as well. Okay. And you say, oh, great. 6 of 7 domains we'll talk about. Awesome. Okay. Now, let's go ahead and move from here and talk about the 6 steps, okay, that it's going to take to carry out this process. So I guess I should say this. So real quickly, okay, in this, you'll notice there's this right here, ACP, and there's this ACP here, okay. It's called acyl carrier protein. So if you remember pyruvate dehydrogenase complex, if you remember, we talked about that there was this arm that came out of E2 and reached into E1, and then it would carry something to E2, and then it would carry something else to E3, and then it would come back, right? The acyl carrier protein is similar to that in the sense that it's going to pick up something and it's going to carry things from one domain to another. That's why it's called an acyl carrier domain, okay? So it's very flexible, and it can move what's attached to this SH group here. It can move whatever is attached to that. It will be an acyl group to one of the other domains that will carry out one of the steps, okay? So all these things are being moved by that acyl carrier protein. Now I'm going to go ahead and skip this slide again because I'm short on time, but this slide just essentially shows you. No, it doesn't show you anything now. So it shows you what the end of that acyl carrier protein looks like. This right here. Okay. So that's the end of the acyl carrier protein. That the rest of the protein, of course, would look like a globular protein. Okay. All right. Now let's go here and look at these steps. Okay. So step 1 here. Okay. Step 1. I'm going to cut this slide in half. Everything on the left would be essentially consistent with what we call step 1 of round 1. You're having a hard time reading that. There we go. Now, step 1 is going to do this. Okay, it is going to load. I'm going to put little quotations around this, KS, with an acyl group. Okay. KS, quote unquote KS, is one of those 7 domains. Okay. It's a synthase, and synthases will carry out condensation reaction, or this one does. Okay. Right, so when we say load KS with an acyl group, that's okay, step 1. Now, it takes a little bit to do this. So I want to go ahead, because this slide, right, what you see here on this slide, there's some things that are implied. So I want to go ahead and talk about this on this next slide. Okay? So what it's going to do, because round 1, we're going to use that acetyl-CoA. So what it's going to do is this. Okay? It's going to take acetyl-CoA, and it's going to take that ACP, acyl carrier protein, and it's going to make what I'm going to call acyl or acetyl ACP. Okay? Meaning this, the acetyl-CoA is going to be attached to that acyl carrier protein. That's the first thing that has to happen. Okay? If you're really curious about this, MAT, okay, That's the domain that carries out that process. MAT stands for malonyl acetyl-CoA transferase. Okay, I believe, yes. Right. And so it's going to essentially transfer that acetyl group to an ACP. Okay, so coenzyme A will come out in this process. Okay. Now, the second step of this, step one of round one is this. We're going to add KS to this. KS is our domain. And to make acetyl KS, okay, plus ACP. Okay. The KS domain is what actually catalyzes that. So the acyl carrier protein picks up the acetyl-CoA from malonyl, or from the MAT, and then the ACP carrier protein takes it to KS, and the acetyl group is transferred to KS. Okay, so what you're left with here is this. Okay, and that is acetyl KS. Okay, that would be step one. Okay. Now, in this case, it's actually an acetyl group, okay, 2-carbon molecule. In round 2, it will be a 4-carbon molecule. In round 3, it will be a 6-carbon molecule, and you can see where I'm going with this. It's going to add 2 carbons every round. Okay. But step 1 will be a transfer of an acyl group. In this case, it just happens to be acetyl-CoA because we're starting it off. Okay. And the next round will be a 4-carbon molecule, though. Questions? Yes?
Like, this is eventually going to be a pretty hydrophobic molecule, so how's this, like, happening in this?
Yeah. Okay, so the question is, this is going to be eventually a very hydrophobic molecule. How is this happening in the cytosolic environment? So I don't know exactly the answer to that, other than to say that that hydrophobic molecule is going to grow on a protein that can accommodate a binding to that lipid, for sure. The next question would be, well, then how is it released and then transported to like the endoplasmic reticulum? And that would be probably some micelle that would have to carry it, or maybe some other protein that would carry it into the ER or something to that effect. Yeah, but there would be hydrophobic portions of this protein around the active site that would allow this thing to grow and be, quote unquote, happy, if you will. Yeah. Okay. We draw it as this kind of like dangling out there, but it's, you know, up against the enzyme. Okay. Other questions? No? Okay. So that's step 1, round 1. There's 5 more steps. Okay. The next step here, let's go ahead and look over on this, on the right side of this. All right. Ringtones. Yeah, I love ringtones. That was a fairly generic one too, you know. Anyway, all right, step 2. Okay, I'm getting distracted. Okay, step 2. There we go. I don't want to get too off topic right now because, you know, I've got limited time. Okay, so step 2. I'll discipline myself. Step 2 now, we start here, again, we have this acetyl group here on the KS domain. That's where we left off down here. The next step, step 2 in all of these rounds is going to be the addition or loading of malonyl-CoA onto ACP. Okay. If you go back a slide or two, remember I said that the whole reaction is going to be 1 acetyl-CoA plus 7 malonyl-CoAs. And they're going to be added together to make this 16-carbon molecule, okay? So we've used our acetyl-CoA already. Now we're using 1 of 7 malonyl-CoAs and attaching it to the ACP. So you can see this. Malonyl-CoA, that's this guy right here, okay? That's added here, as you look at this, added to our ACP, okay? MAT also does that. Okay. So MAT loads either acetyl group onto ACP or a malonyl-CoA onto ACP. Okay. Right. And that's step 2. And what you're left with at the end then here is a acetyl group on KS and a malonyl-CoA on ACP. Okay. Now, I'm going to go ahead and draw the next 4 steps, and just write them out here on this blank slide. Okay. The next step after this would be step number 4, is what would be considered a condensation step. Okay. Step number 3, and this is going to be catalyzed by KS. I will show you the details of that here in a moment. Okay. So that's condensation. Go ahead and zoom in on this. Condensation. After condensation is going to be a reduction reaction. Okay, that'll be step number 4, and that will be catalyzed by Kr. If you're looking for the subunit Kr. Kr is a reductase. The R stands for reductase. K something else. Okay, you can go back and look at it on a slide. Okay. After this reduction, there will become a dehydration. Not dehydrogenation, but dehydration. This is going to be catalyzed by DH, dehydratase, not dehydrogenase. Okay, so be careful in this. Okay, this is a dehydratase. It's going to remove water. Okay, dehydration. That's step number 5. And then finally, the last step is another reduction reaction. Which is going to be catalyzed by ER. Again, the R stands for reductase. Okay. Now, interestingly, and I'm going to show you a slide of this here in just a second, okay, but interestingly, those reduction reactions are occurring on what would be considered the beta carbon. So this process, while we call it lipid biogenesis or lipid synthesis, we could also think about it, and I've never actually seen this term used in a textbook, but I think it would be appropriate, it would be beta reduction. Okay, because it mirrors beta oxidation. In fact, it's the exact opposite. So if we're to look at beta oxidation starts with an oxidation reaction, okay, catalyzed by acyl dehydrogenase. From there, it goes to hydration. Okay, so this would be, if you're looking at beta oxidation, this would be step 1 and step 2. From there would be another oxidation reaction. And then from there would be that cleavage step where we didn't really mention this, but it's really a hydrolysis step. Hydrolysis is the opposite of condensation, okay? And that would be step 3, step 4. Okay, so beta oxidation is going in this direction. And like I said, I've never seen this term used before, but we could think about going in the opposite direction, beta reduction, okay? And they're mirroring them. Okay, so if you think about this especially from a studying standpoint, this is pretty easy to study. When you get one of them down, you just think, okay, it's just the opposite, going in the opposite direction. Okay. Obviously, there are differences. Now, let's go ahead and move from there. I want to show you those steps 3, 4, 5, and 6. Okay, I'm going to skip. No, this is the slide I want to use. Yeah, I'm going to go ahead and. Do I want to use that slide? Yeah, we'll use that slide. Okay. Don't worry about the numbers. The numbers, I'm going to get rid of. I'm using a different numbering system. Okay. All right. So we're starting here now. What we have, you'll notice this is after the second step. So we have now come back up here. Right, this guy right here, we have that on the bottom right-hand corner, we have that malonyl ACP, and that KS acetyl group. Okay, so that's really what we are starting with right here. Okay, you'll notice here as we go through this, this is that third step, condensation. CO2 is broken off in this, and now we have, it's really, that will be converted to HCO3 through a couple of processes in the cell. Okay, that's our condensation step. Again, KS does that. Okay, next step, right? Real quickly, I guess I should highlight some of the details of this. You'll notice in this condensation step, these electrons here, let me go ahead and zoom in on this. Okay, these electrons are going to nucleophilically attack the acetyl group. Okay, these are electrons that are going to come here, and there's going to be essentially a nucleophilic substitution reaction. And what you're left with now, sitting on the ACP, is a 4-carbon molecule. And here's our beta carbon. ACP, remember, is real flexible. So it can now carry that 4-carbon molecule to something else. And that's exactly what it's going to do. What it's going to do next is this, a reduction step. Catalyzed by, it will take this to, from KS, it will take it to KR. Okay? And you'll notice here, that carbon, the beta carbon, there's a ketone up here, now it's an alcohol here. It's a reduction step. Adding electrons to that carbon. NADPH is going to be used as the electron donor in this case. Okay? And you're left with this alcohol, 4-carbon alcohol. Now come over here, so zoom out real quickly. Come over here, and that alcohol is going to be dehydrated. I'll remove water from this, okay? And that's going to be happen by a dehydratase, but we go from alcohol here through dehydration, water is going to come off, and we're going to get now a trans double bond. That will be catalyzed by the domain DH, dehydratase. Okay. We are now a single step away from having a saturated 4-carbon acyl group. Okay. We will see then one last reduction step, catalyzed by ER, and that beta carbon now has been fully reduced. NADPH is once again our electron donor. So every round requires 2 NADPHs. There are 7 rounds, which means that you'll have to have 14 NADPHs, which is the number I gave you. Okay. Every round will consist of these 6 steps. Okay. Loading of an acyl group onto KS, loading of a malonyl CoA onto ACP, and then condensation, reduction, dehydration, reduction. Okay. Right. Questions on that? Yeah.
Actually, is there like species that will basically stop at those points before they ever actually get to the fatty acid?
So,
Just take it off?
yeah, so that's a good question I thought about on the way here and didn't have time to look it up on the way here. The question, if you didn't hear it, is there going to be some fatty acid synthases that will stop at some shorter chain than 16 carbons? And I don't know the answer to that. I mean, we do have precedents for shorter chains.
Like in the pathway itself, like, so it's making alcohols and whatnot, so isn't it?
Oh, cleave those off? As byproducts, you mean, right? So so instead of going through the whole dehydration reduction stop at the alcohol, it's possible. I don't know the answer to that question either. I thought you were asking, could you make a 14 carbon rather than a 16 carbon? And I don't know the answer to that either. It's possible that we make a 16 carbon and then things are broken off. I mean, that's the way biology does things sometimes, but I don't know. Other questions? Okay. All right, let me go ahead and then I'm going to skip that slide, right? But let me just kind of show you this. Okay, so we just ended here, okay, where we had this molecule, this 4-carbon molecule, but it was on ACP. Okay, this would be, so if you start to look at your next round, that 4-carbon molecule will be transferred in the step 1 of the second round. That 4-carbon molecule will be transferred to ACP. And then step 2 will take on another malonyl-CoA on the ACP. So now you have 4-carbon molecules sitting on, this would be KS. Right there. And then you have malonyl-CoA sitting on ACP. Okay. And then you'll start the 4 rounds again. Okay, so step 1 and step 2 are implied, but that you would get there, right? But then you go condensation, reduction, dehydration, reduction, and then you would have to transfer in step 1 of round 3, transfer a 6-carbon molecule from ACP to KS, load a malonyl-CoA in step 2, and then condensation, right? Reduction, dehydration, condensation. Transfer 8, and that's what this is just showing. Okay, and there will be 7 rounds of that. Okay. Step 1 and step 2 of the next round are not really shown here; they're implied here. Okay. Yes.
Is it the transferase that moves it back to the KS?
Yeah. Yeah. Okay. So the question is, it would be the transferase that does that. Yeah, it'd be MAT every time. Yep. Okay. Other questions? Actually, I just thought of what you were asking. No, it doesn't. You were asking about the 4-carbon molecule? Yeah, it's actually KS that does that.
Okay.
Yeah, so KS takes the 4-carbon molecule off of ACP, puts it on itself. MAT will load the 3-carbon malonyl-CoA onto ACP. Okay, so it's only that. It's the only time that MAT is used to load ACP with the acyl group is that first round, and it's a acetyl group. Okay, I'll give myself a red check minus. Some of you are wondering, he's got to give himself a red check minus for that. I get you. Yeah. Other questions? You'll end with this. Okay, fatty acid synthase will produce a 16-carbon molecule. That can then go on and move into the ER and become an 18-carbon molecule. Okay, it can, in the ER, it can also be exposed to desaturations, which can then take it from fully saturated molecule to unsaturated, polyunsaturated, possibly introducing cis double bonds. Okay. We're not going to look at that, but that would be this next slide and then several slides after that. Okay, we're going to stop right there. So I'm going to put a big red X right through this, and then every slide after that we're not going to go over. We need to move to amino acids. Yes.
Does it always make the palmitate the 16-carbon saturated fatty acid, or is that just the one that we're talking about?
Well, so it always will make the 16-carbon. Right, so that was kind of the question that was asked earlier. Does it make that? So it's my understanding that it always makes the 16-carbon. Okay, it's possible that it could stop and make a 14-carbon, but my intuition tells me that actually make the 16-carbon and then something else in the ER would reduce the number of carbons in that. That's typically how these things work. This will just drive through more than likely and make the 16-carbon molecule and then stop at that, and then other enzymes will be used to modify it after that. Okay. Let's do this in the interest of time because we're running very short on it. I want to move to amino acid metabolism. Okay, so amino acid metabolism, we're going to talk first of all about absorption. In terms of, and transport in particular. Transport's going to be really important of amino acids as we get into this. Okay, so we're going to look at some of that in the next couple of days. We will talk about something called the urea cycle, which is central component of amino acid metabolism. Hopefully we'll talk about that on Wednesday. And then we'll see what we have left on Friday to talk about. Okay, we'll hopefully get to some oxidation of amino acids. Amino acid oxidation and synthesis is complex because there are 20 different amino acids. Okay, so there are common themes in those things, but certainly we're not going to go over 40 different pathways. Not in the next 3 days. Some of you can thank me later. Okay, so let's go here and let me go ahead and do like a comparing and contrasting to what we have done. At this point in time in the class, we have now talked about carbohydrates and we have talked about lipids. Okay, we have seen how those can be broken down and also made. The interesting thing about these is that they are comprised entirely of CHO, right? Carbon, hydrogen, and oxygen. If we were to look at the breakdown of these, they are broken down into CO2 and water, and of course the energy that can be harvested from them, as we have discussed. Nothing new there. We can store these molecules. Carbohydrates can be stored as glycogen. Lipids can be stored as triacylglycerides. Okay, glycogen in skeletal muscle, glycogen in the liver, triacylglycerides in hopefully adipose tissue and some other tissues as well. Okay, but we can store them too. Right? Now, when you get to proteins, there is some notable differences. Okay, first of all, there's no storage. Okay. Every protein that is made in our bodies is made with a particular function. So when we start to have to harvest amino acids from the body to do something from an energy standpoint or an energy needs, it's harvesting protein that's functional. Okay, for instance, your muscle cell is skeletal muscle is a great resource for protein and amino acids. So in starvation stages or even in fasted states, your body will start to break down protein in your skeletal muscle. Okay, to derive, and we can get from that amino acids that can be used to make things like lipids and in particular carbohydrates, glucose. Okay, 18 of the 20 amino acids can be used to make glucose. So very important from that standpoint, but we don't store it. Okay, there's obviously a ton of protein in our body, but we're not storing it, we're using that protein. Okay, the other thing is this, and this is going to be a really important factor as we look at this. Certainly, amino acids and proteins are made of carbon, hydrogen, and oxygen. However, importantly, every single one of them also has a nitrogen in it. Okay, so that means that these, when we break these down for energy needs, CO2 and water will be made plus nitrogen, which is going to be put in a molecule called urea. Okay, and urea is this molecule, and we will look at how this molecule is made in the liver. Incidentally, the liver is the only place that this molecule is made. I'm trying to draw it in, and I'm having a hard time. There we go. Okay, so that's urea. Now here's the problem with the nitrogen. Okay, it can be toxic to us, and certainly smaller concentrations than like carbon dioxide would be toxic to us. Okay, that can be toxic as well, but the nitrogen can be toxic. So the body has got lots of ways to deal with nitrogen and how it's going to transport that nitrogen, in particular in the blood. So we're going to think about some of that. Okay, now generally speaking, as we look at this, we're going to be looking at how proteins will be broken down into amino acids. These amino acids, okay, can be broken down into alpha keto acids. Alpha keto acids, to remind you, I've mentioned this before, but I'm going to go ahead and mention it again. An alpha keto acid is this. Okay? And there are several of them that we've encountered in this class. For instance, pyruvate is an alpha keto acid. Oxaloacetate is an alpha keto acid. Alpha ketoglutarate is an alpha keto acid. Those three comprise the most common alpha-keto acids that you will find in the cellular system. And amino acids, when they're broken down, will usually land at one of these 3 points: pyruvate, oxaloacetate, or alpha-ketoglutarate. Some of you are looking at that and saying, that sounds like the malate aspartate shuttle to me. Exactly. Okay, I told you that was going to be important long ago, okay, and it keeps coming up to enrich your lives. Anyway, some of you say, no, it's haunting me, honestly. I'll be real honest with you, okay? Now, as we look at this, of course, you'll notice that the alpha-keto acids, none of them have nitrogen in them. Okay, which means that going from amino acid to an alpha-keto acid means that the nitrogen has been removed. It can be removed directly as an ammonium cation, which is very toxic. Okay, so we're going to look at how that's dealt with in the cell. Or it can be removed through what's called a transamination reaction. Okay. And transamination reactions are really, really important. Okay? Transamination simply means we're going to take an amine group off of an amino acid, we're going to put it onto another alpha-keto acid and make another amino acid. So we'll see how these things are transferred from one amino acid to an alpha-keto acid to make another amino acid. Okay? Eventually, though, that nitrogen is going to have to come off as an ammonium cation. Okay? But that's regulated in terms of how that happens. Okay? Right, so that's an introduction to amino acid metabolism. Right? The main point of all of this is going to be to monitor nitrogen. Where is that nitrogen going to be moved to, and how is it going to be dealt with? Okay? All right. Now let's do this. We're going to skip this slide. You eat protein-rich meal. It goes into your gut. Proteases cut it up. You absorb amino acids. All right. Awesome. Some of you are saying, well, I wish all of our slides could be like that. No, you don't. You really don't. You'd feel gypped if it was. Okay. All right, but that's what's going to happen. Now, amino acids, when they get into the small intestine, are going to be absorbed in an identical fashion as carbohydrates. So if you remember, glucose was absorbed into the intestinal cell using a sodium gradient. That sodium gradient was established by a sodium-potassium ATPase. Right? Amino acids are the same thing. We have these symporters, okay, that move amino acids and sodium into the small intestine. The amino acid then can be dumped into the blood vessel, okay? And that, of course, is our endothelial cell that's doing this. That sodium-potassium ATPase, or that sodium-potassium pump, is really important in this. I'll reference you back to your notes on carbohydrate absorption if you want to, further your understanding of that, or just remind yourself of what that looks like, okay? Same idea, though. Now, you will notice here, and this is going to be true, it was true of carbohydrates, it's true of this, that this is here all of the blood vessels from the large intestine, all the blood vessels from the small intestine, all of the blood vessels from the stomach, all dump into this very large vein called the portal vein, and that portal vein goes right into the central portion of the liver. Right? So every amino acid that is absorbed into our bodies, it gets dumped into the portal vein, and the portal vein then carries those to the liver. The liver is central to the metabolism of proteins and amino acids. Okay? And we're going to see that time and again. Okay? But keep that in mind from an anatomical standpoint, that's where all the blood's going to go. Okay, from a meal. Now, we're going to look at what I want to do in the next 5 minutes. I won't get through all of this, but what I want to do is talk a little bit about what is in that blood as it reaches the liver. Okay, and where does that come from? Okay, so let's say this. Let's start here. The amino acid glutamine. Okay? 70% of the small intestine, okay, energy needs are met by glutamine metabolism. Okay? Interestingly, it is the preferred fuel source for the small intestine. I mean, it can use other things for sure, but it uses glutamine to meet the majority of its needs, energy. And there's a lot of energy need for the small intestine because it has to run that sodium potassium ATPase all the time. Okay? So there's a lot of it. Now, how is it going to get that? Well, it's going to do this. There is this enzyme in the small intestine called glutaminase. And glutaminase is going to deaminate the side chain of glutamine. So it's going to remove that nitrogen. Now, don't worry about this. We'll get to that later on. Okay? So it's going to remove it as an ammonium cation, and it's going to put that into, right, the blood vessel that will be taken to the liver. That will make glutamate when it does that. So glutaminase deaminates the side chain of glutamine, making glutamate. Glutamate will undergo a transamination reaction. Okay, in this case, pyruvate, I guess I should write it down here. Okay, he's going to take the other nitrogen here, and it's going to make alanine. So pyruvate, remember, is an alpha-keto acid. It's an amine group away from becoming alanine. This is a transamination reaction. That will make alpha-ketoglutarate. That then goes to the citric acid cycle, oxidative phosphorylation, and that, of course, makes ATP. That's how the energy needs of the small intestine are going to be met. Glutaminase deaminates it, a transamination reaction, what makes alpha-ketoglutarate, and then we're on to the races with citric acid cycle and oxidative phosphorylation, which we've already talked about. Okay? Now, real quickly, okay? Transamination. I need to mention this because that's the first time we've come across transamination. We're going to come across transaminations a lot in this class, right? There is a host of enzymes, multiple forms of them, called aminotransferases. All of them catalyze this transamination reaction. Every single one of them will use either glutamate or alpha-ketoglutarate, right? So this is found in all of these. Okay? It can go in either direction, back or forth. Okay? They'll either use alpha-ketoglutarate to make glutamate, or they'll make alpha-ketoglutarate from glutamate. Okay? One or the other. They can then use any other the other amino acids, and again, it can go in either direction, to make an alpha-keto acid. This would be oxaloacetate, pyruvate. Those would be the two most common. Not alpha-ketoglutarate, because it's already using alpha-ketoglutarate. Okay. So that's a convenient way to shift the amine group from one amino acid to another. Alanine, as we're going to find out, alanine, glutamine, and glutamate all are really central to this whole thing. Okay? Aspartate plays an important role in it as well. Okay? Those are the most common types of amino acids that we find carrying nitrogens. Okay? Right. Now, here's what we need to do. Okay? Next time, let me say this. The amount of ammonium cations that's coming from the bloodstream in from our small intestine is enough to kill us if it's not filtered through the liver. Okay, so we're going to look at how the liver is going to filter that ammonium cation starting on Wednesday, and we'll complete the urea cycle on Wednesday. Okay? All right, I will see you on Wednesday. Have a good rest of your day.